Yes, our next speaker is also the last one before we come to lunch. It's Patricia Goncalves, and she'll be talking about horse mackerel, I think, different types of horse mackerels. So Patricia, the title of your talk is How Different Are the Eggs of Trachurus and Trachurus picturatus, Implications for EPMs and in the North Atlantic waters. So the floor is yours, and I see you don't need help with the computer, so just go ahead.
Good afternoon to everybody. The aim of this study is to answer this question: how different are the eggs of Trachurus and Trachurus picturatus? In Iberian Atlantic waters, ICES Division 9A, we have 3 species of the genus Trachurus: horse mackerel, Trachurus, blue jack mackerel, Trachurus picturatus, and the last one, Mediterranean horse mackerel, Trachurus mediterraneus. Both of them have almost the same length at the first maturation and the same age. The Mediterranean horse mackerel is the less abundant of our region. That's why these data are not from the Iberian waters. This is just to take a look. This is the 3 species. The first on the top is the horse mackerel. In the middle is the Mediterranean species with the yellow tail, and the last one is the blue jack mackerel. You can see the more darkness in the dorsal part of the body, but they are really difficult to distinguish. According to their distribution, we can say that the Trachurus and Trachurus picturatus have the same distribution area and are more or less abundant. But of course, Trachurus is the more abundant species for our region. The less abundant, as I said before, is Trachurus mediterraneus. You can see here just on 3 parts of the ICES Division 9A. Okay, the most abundant ones have also the same main spawning season, what could be a problem for us. According to egg characteristics, if it's difficult sometimes to identify the adults of the 3 species, imagine on eggs. Okay, according to literature, Trachurus eggs is described as being pelagic, spherical, and have the range diameter between 0.8 until 1.04. They have a single globule with the range between 0.19 until 0.29 and segmented yolk. According to Trachurus picturatus eggs, there are no references about their characteristics on the literature. You know all that egg identification in plant samples is needed for purposes of stock assessment, mainly the application of the daily egg production method. This is the general formula of the daily egg production method. Is this P with the red circle is the egg production. If we are classifying of our eggs of Trachurus picturatus as being Trachurus, we are overestimate our egg production and of course overestimate also our spawning stock biomass for horse mackerel. This is our last DPM application for horse mackerel. On the left side, you have our grid of plankton sampling. For planting sampling, we use 2 nets, Calvo and Bongo, and we also use Kufish. But Kufish is just to give us an indication during on board to make some decisions. Because when we are sampling offshore in the stratus, if we get 2 negative Kufish with no eggs of no species, we can change strato. This is our information about our adult sampling, our trials, and you can see here that on 5 stations we catch also Trachurus and Trachurus picturatus, and in one of that we catch the both species spawning. And we see here a good opportunity to make some fertilization experiments to try to find if there are differences between the eggs. This is the basic information about the fish that have been captured in that station. You can see on the B is the data referred to Trachurus picturatus. We just catch 2 spawning females and use that to make the fertilization experiments. This is general information about our experiments. We use the temperature of 16.5 because it's more or less the temperature of the water in that trough position. And we, in variable intervals, we take some eggs off the tanks, we fix it with formaldehyde, and the analysis and description of morphology has been only made on the laboratory. We have to say that when we come to laboratory, we have a surprise with our Trachurus eggs because they don't develop, and almost of the data that the eggs we have been collected are in stage 1 because they died. That's why for this comparison you use the collection data of the eggs of a previously artificial fertilization for 2004 and already been published to compare the development of the both species. Okay, this is our first stage of egg development for the both 2 species. We can see here the pointer. Here is when the division starts. And we can see that the Trachurus picturatus has also a segmented yolk. Here the oil globe seems a little bit bigger than on Trachurus, but I have to say that it's broken. This is the blastula phase. Once again, we don't see so much differences between these both species. Here we have just now the embryo phase, and you can see the beginning of embryo's development, with the tail lies against the polar region of the yolk and continues the line of the yolk mass, and until now no differences. Now, on this stage, the pigmentation is already visible. We have on both species pigment spots form 2 rows along the dorsal body of the embryo, and is common on two species. In this stage of development, the tail is bigger than 1/4 of the yolk sac length, and tail curvature is evident. On these images, we can see the pigmentation on the head of the 2 species. And again, there are no differences. And this is the last stage, and they are almost watching, and no differences. Okay. According to morphology, we cannot see any differences. And about sizes, we determine the sizes of our fertilized eggs. And this is for Trachurus, this is the description of the literature, and we can say about our Trachurus picturatus, based on our experiment, that the cordon diameter ranges between 0.98 until 1.1, and oil globe 0.19 until 0.31, and they have also segmented yolk, but the size overlaps between the 2 species. Here we have the number, these pots represents the number of eggs in each stage at each sampling time. At the lab, we classify each egg into an 11-stage scale key, according to the descriptions of Cunha et al. Of 2008. Here you can see how to convert the 11-stage scale key into the 4-stage key used by the WGMAGs. You can see that for this species you have continuous development. And eclosion occurs 75 hours after fertilization. Okay, this is our Trachurus from 2010. That what I say to you, you miss some stages because we catch eggs that aren't developing. And eclosion, but we get some larvae also. And the eclosion occurs 65 hours after the fertilization. Just to have a look for the Trachurus data of 2004, it's the same development, a continuous development along the time, and the eclosion is around 60 hours. Okay, just to see if there are differences on the durations of stages between the two species. We applied the multinomial model to see the time duration of each stage of development and try to compare between the two species. Somehow, you don't know, but Trachurus eclosion occurs earlier than on Trachurus picturatus. And according to this data, it seems that until the 30 hour of sampling, all species are developing in the same way. But after that, the stage 6 and 7 for the Trachurus, it's more quickly on develop. And that's why the eclosion is so close to the 60 hours after fertilization. And at this moment, that's all we have. And we can conclude for now that egg identification between those two Trachurus species depends mostly on the size of the chorion and all globe diameter. However, we see before that size is not enough due to the overlapping. And also, according to the bibliography, egg size may vary within a species, between populations of the same species, and among and within species. Being those morphological characteristics insufficient for species identification, we think that genetic is the future, should be applied, because until now, this is an egg from our DPM survey. We can say or assure that this is Trachurus egg or Trachurus picturatus. And for validate our data, we must apply genetica. Okay, and that's all. Thank you very much for your attention.